Hello friends, good evening. Welcome to Sam Biology class. Friends, today we shall discuss about glycogenolysis. Before discussing, I request all of my friends, please subscribe my YouTube channel. Then what is glycogenolysis? It is the biochemical pathway which breaks down glycogen into glucose 1-phosphate and glucose. The chemical reactions occur mainly in the liver as well as in the muscle. The process of glycogenolysis is under the regulation of two different enzymes, phosphorylase kinase and glycogen phosphorylase. Friends, you know that glycogen is a branched homopolysaccharide of alpha D glucose units. It is the principal storage carbohydrate in animal. Glycogen synthesis occurs mainly in the liver as well as in the muscle. Liver glycogen play a very important role in maintaining the normal blood glucose level in fasting state by the process of glycogenolysis. By glycogenolysis, liver glycogen is broken down into free glucose units which are then released into the bloodstream, thereby maintaining the normal blood glucose level. Whereas muscle glycogen is cleaved into glucose 1-phosphate which is then oxidized by the process of glycolysis, thereby providing energy for muscle contraction. There are two different enzymes that participate in the process of glycogenolysis. They are number 1, glycogen phosphorylase. Glycogen phosphorylase. This enzyme catalyzes the cleavage of alpha 1,4 glycosidic linkages sequentially from the non-reducing ends of a glycogen molecule yielding glucose 1-phosphate. The second enzyme is debranching enzyme. It is known as debranching enzyme. It is one of the few known protein which has two different catalytic activity that are located on the same polypeptide chain. So debranching enzyme is a bifunctional enzyme and the different activities are transferase activity and the other is alpha 1,6 glucosidase activity. So by the combined action of glycogen phosphorylase and debranching enzyme, a complex macromolecule such as glycogen is cleaved into glucose 1-phosphate and glucose in the ratio of 8:1. Friends, glycogen is a branched polymer of alpha D glucose units. Actually, it is a large molecule. This figure shows the reaction steps of glycogenolysis, and it is the part of a glycogen molecule having two types of glycosidic linkages. The linkages which are present at the branch point are alpha 1,6, whereas the remaining all glycosidic linkages are alpha 1,4. First of all, the enzyme glycogen phosphorylase catalyzes the cleavage of alpha 1,4 glycosidic linkages sequentially from the non-reducing ends yielding glucose 1-phosphate. This process, called as phosphorylasis, continues until 4 glucose residues remain on either side of the branch point. The sequential removal of glucose units from the non-reducing ends result in the formation of a much branched core polymer known as limit dextrin. The second enzyme that is involved in glycogenolysis is debranching enzyme. As already mentioned, it is a bifunctional enzyme. It has transferase activity and alpha-1,6 glucosidase activity. First, by its transferase activity, debranching enzyme catalyzes the transfer of a segment of 3 or 4 glucose residues attached at the branch to another chain, thereby forming a new alpha-1,4 glycoside linkages. In the next step, the same debranching enzyme by its alpha-1,6 glucosidase activity cleave the alpha-1,6 glycoside linkages, thereby yielding free glucose residues. So the cleavage of glycosidic linkages by glycogen phosphorylase and debranching enzyme resulted in the formation of this glucose chain, which is further cleaved by the action of phosphorylase. So, in the process of glycogenolysis, by the combined action of glycogen phosphorylase and debranching enzyme, a large branched molecule such as glycogen is cleaved into glucose 1-phosphate and glucose in the ratio of 8:1. After that, glucose 1-phosphate is further converted into glucose 6-phosphate by the enzyme phosphoglucomutase, and the fate of glucose 6-phosphate depend on the tissue where it is formed. In liver, kidney and intestine, there is an enzyme called as glucose 6-phosphatase. Glucose 6-phosphatase catalyzes the hydrolytic removal of the phosphate group of glucose 6-phosphate, thereby yielding free glucose units. So, by the process of glycogenolysis, liver play a very important role in maintaining the normal blood glucose level in fasting state. Whereas in other tissues such as muscle and brain, the enzyme glucose 6-phosphatase is absent, so free glucose units are not formed. So friends, this is all about glycogenolysis. For more updates, please subscribe Sam Biology class. Thank you very much.